Hi, it's Jen and welcome to my channel, and in today's video I'm going to be talking about the neuroscience of circadian rhythms. Circadian rhythms are physical, mental, and behavioral changes that follow a daily cycle. They respond primarily to light and darkness in an organism's environment. Sleeping at night and being awake during the day is an example of a light-related circadian rhythm. As someone who has struggled with insomnia in the past, I've always wanted to learn more about the neuroscience of circadian rhythms, and I also know that disruptions in the circadian rhythms are related to bipolar disorder, which I'm also very interested in, so in future videos I'll probably want to talk about that. But in today's video, I'm just going to focus on the basics of the neuroscience of circadian rhythms. So our circadian rhythms are controlled by two main things: our internal pacemakers, which I'll talk about the neuroscience of later, and external factors such as the light and dark. And in circadian rhythm science, they call that a Zeitgeber, I think is how you say it. So it's just whatever stimuli affects you the most. So in our case, we are very dependent on our sight, so the light and dark is our main Zeitgeber. But for an animal that doesn't use sight, it might be a smell or sound or a different sense that is the Zeitgeber for that animal. When we are placed in a situation where there are no light cues, so it's either light all the time or dark all the time, our internal pacemaker actually has us have a sleep and wake cycle that is actually 24 hours and 9 minutes long on average. So for some animals it could be 25 hours or around that. But what's interesting is that it's not exactly 24 hours. So if you were placed in the dark for months at a time, each day you would probably, if you were like the average human being, you would shift by 9 minutes from the normal day every day to eventually you might be totally awake at everyone else's nighttime and asleep during the daytime. So it's shifting. That proves that we aren't fully dependent on this light and dark cycles because we have our own cycle of 24 hours and 9 minutes. However, when we are out in the real world and we are responding to the light and the night, our cycle is not 24 hours and 9 minutes because we are affected by the light and affected by darkness, and I'll explain the neuroscience of that later. To understand this better, we will look at this graph, which is called an actogram. The actogram represents the activity of a diurnal bird, that is a bird that is awake during the day and asleep at night. The x-axis represents the time in hours as labeled here at the top of the graph. So 0 hours, 24 hours, and 48 hours. At the top of the graph, you can also see what the lighting was like during the different days. So for days 1 through 6, you can see it was dark all the time. For days 7 through 39, you can see that there was 12 hours of darkness, followed by 12 hours of light. For days 40 through 77, you can see that there was still periods of darkness and light, but the light periods were longer than the dark periods. And then the same for days 80 through 17. And then through days 118 through day 155, it was dark all the time again. Let's start by looking at days 7 through 39. So this section right here. On those days, the birds were exposed to 12 hours of light and 12 hours of darkness. The black dots here represent when the bird is active. So you can see that the activity of the bird aligns with the light period shown here. And you can see when the light is off in the dark period, there's essentially no activity from the birds, as you can see here. For days 40 through 77, which is this section right here, we can see that again the birds are active when the light is on and inactive when the light is off. So here and here. And on those days, the light was actually on for 18 hours at a time and off for 6 hours at a time. So with extended light, there is more activity. In both cases, the cycle repeated every 24 hours, but the amount of light and the amount of darkness was different. And you can tell that these cycles are 24 hours because each day, their activity begins about at the same time within these cycles. So there's that straight line here, whereas when there aren't on 24-hour cycles, there's like a shift every single day, and there's not a straight line, as you can see for days 1 through 6 and through days 118 through 155. So day 80 through day 117, which is this part right here, is basically the same as days 40 through 77, in that the light was on for 18 hours and off for 6, but the time the lights were on were shifted. And as you can see, the activity shifted along with the shift in the time the light was on. So you can see here that they shifted when the light started and when the dark started, and the behavior lined up with that shift. So for days 6 through day 118, we see these straight lines and these 24-hour cycles because the birds are matched up, their behavior is matched up with the light. And when that happens, that is a phenomenon called entrainment. So when entrained, you wake up every day at the same time and you're aligned with the light and dark cycles, such that for diurnal animals, like us as well, they're active during the light and inactive during darkness, and the reverse is true for nocturnal animals. So as I mentioned earlier, when there is a stimulus, such as a change in light or a change in darkness, that is called a Zeitgeber. So each of these points, when light goes to dark or dark goes to light, that is an example of Zeitgebers. So days 7 through 117 all have Zeitgebers, whereas days 1 through 6 and days 118 through 155, since it's dark all the time, there's actually no Zeitgebers. And this would be true if it was light all the time too. Those would be instances where there's no Zeitgebers. Next, we will look at days 1 through 6 and days 118 through 155. During these days, the lights were off at all times. If activity was controlled solely by light, then there would be no activity during these times. But you can see here that this is not the case, as there is still plenty of black dots that represent activity. As you can see here, here, and up here. Instead, we see that each day, the cycle of activity and rest is greater than 24 hours. We can see this because each day, activity starts and ends later than it did the day before. Subsequently, the periods of rest also start and end later than they did the day before. So as you can see here, about on day 120, it's starting later and ending later than it did on the day before. And same with the rest. The rest is starting later and ending later than it did the day before. So in these cases where it's dark all the time and there's no Zeitgeber, this is called the free-running rhythm, and that's the rhythm that's controlled solely by the internal pacemaker of the animal. So this cycle that's greater than 24 hours is caused by the internal pacemaker of the animal, and it could be called the animal's own circadian clock. For this clock to function properly, it must be reset daily by the Zeitgeber of light or darkness. The circadian timing system, or CTS for short, integrates the internal pacemaker with external cues from the Zeitgeber. The CTS consists of photoreceptors and entrainment pathways that provide photic information to the pacemaker, and pathways that extend from the SCN that affect systems under circadian control. To start, I will show the neuroscience of the photic entrainment pathways that lead to the pacemaker. So the pacemaker is the suprachiasmatic nucleus, which is located here in the hypothalamus in this picture, and right here in this picture. I want to point out the optic chiasm, which is right here, and you can't see it here, but in the second picture is right here is the retinohypothalamic tract that projects from the retina directly to the SCN. When all visual pathways leading to optic chiasm are cut, it leads to an animal with no visual reflexes, but a perfectly normal entrainment of the circadian rhythm if the pathway from the RHT to the SCN is intact. So this means that an animal could be completely blind, but still be entrained and in sync with the light and dark cycles, which I find to be very interesting. On the other hand, if the optic chiasm is intact, but the RHT is severed, an animal could have perfect visual eyesight, but not be entrained to the light and dark cycles. In many cases of blindness, however, both areas are affected, so many blind people are in fact not entrained. To get a better understanding of where exactly the SCN is, we have to realize that it is in the hypothalamus, which is located here in the brain. And when you zoom in on the hypothalamus, the SCN is right here. There are 4 lines of evidence that establish the SCN as the dominant mammalian circadian pacemaker. Firstly, as we have seen, the SCN is the major site of termination of the photic entrainment pathway, the RHT, and it also receives other entraining input such as the movement from the organism. Lesions in the SCN result in a loss of nearly all circadian rhythms. SCN ablation abolishes the sleep-wake rhythm, but the total amount of time spent awake and asleep and the proportion of REM and slow-wave sleep are unaffected. So you can see in this experiment that it was dark for 24 hours. And when the SCN is intact, you can see that the cycle has a period of activity and then a period of inactivity. And you can also see that the cycle is greater than 24 hours because each day the activity starts a little bit later each day. Arbitrarily, we could say that the cycle here is 24 hours and 9 minutes, just like it is for humans. When the SCN is lesioned, there is no 24-hour and 9-minute cycle. The cycle is random, and there's periods of activity and inactivity right after each other, and there's just no rhythm involved anymore. Thirdly, the SCN exhibits a circadian rhythm in neuronal activity even when isolated. And lastly, transplantation of fetal SCN into the brains of animals rendered arrhythmic by SCN lesions restored rhythmicity. So that is, if a fetal SCN was transplanted into this subject right here, the rhythmicity as above would be restored. So it would start looking like it does in the top in this bottom part. Lastly, we will discuss the neuroscience of the pathways that extend from the SCN that affect systems under circadian control. It is also important to note that the SCN exhibits a rhythm in neural firing with a discharge rate that is high during the day and low at night. So you can see here that the SCN projects to the forebrain and the thalamus, which in the end results in attention, psychomotor performance, memory, sensory-motor integration, and emotion. And it makes sense that these would all be more active during the day than at night. The SCN also projects to the hypothalamus, which results in regulation of body temperature, endocrine regulation, autonomic regulation, metabolism, sleep-wake cycles, and melatonin. So this is just a broad overview of how the SCN affects our behavior through its projections to the different areas in the brain. Now that we know more about how circadian rhythms work and the neuroscience behind circadian rhythms, I look forward to exploring them more deeply in future videos. As I mentioned earlier, I'm really interested in how circadian rhythms affect bipolar disorder, and I'd also be interested in understanding how things like over-the-counter melatonin affect the circadian rhythms, and any research done on blue blocking glasses and things like that, because I hear about them a lot and I'm curious to know what the research really says about those things. I'm also open to any suggestions or questions that you might have, so be sure to leave a comment below to let me know. If you like this video, please like it and subscribe if you want to see more from me. Alright, bye!